Prostate cancer is the second most common cancer in men worldwide, accounting for hundreds of thousands of new diagnoses every year. This disease operates on a unique timeline. Precancerous lesions enter a long latency phase, taking 10 years or more to develop into invasive malignancy. During this window, these cells rely on specific androgens, primarily dihydrotestosterone, or DHT, their essential growth fuel. This decade-long reliance on a symbol hormone suggested a path for chemoprevention. Researchers hypothesized that by using a daily pill to block DHT, they could starve these lesions before they ever became a threat. These theories led to some of the largest clinical trials in medical history. However, the data gathered from these studies challenged the fundamental assumptions of urologists. The search for a simple preventative measure revealed a biological conflict. In this case, applying a logical intervention resulted in a series of counterintuitive clinical outcomes. One major attempt at prevention was the SELECT trial, monitoring over 35,000 men to determine if these supplements provided a protective shield. The study was terminated years ahead of schedule. An independent safety committee reviewed the preliminary data and ordered an immediate halt to the trial. Vitamin E supplementation did not lower cancer rates. Instead, it led to a statistically significant increase in prostate cancer diagnoses. Selenium showed similar results. In men who already had high levels of the mineral, supplementation increased the risk of high-grade aggressive prostate cancer. These results suggest that the prostate health supplements found in most pharmacies are backed more by marketing than biology. Without hard clinical evidence, these interventions can move from ineffective to active harm. Researchers also investigated 5-alpha reductase inhibitors like finasteride. The PCPT trial was designed to see if these clinical-grade hormone blockers could succeed where vitamins failed. By blocking testosterone conversion, the drug achieved a 25% reduction in the overall incidence of prostate cancer. However, the trial data contained a concerning correlation. The group taking the drug also showed a statistically significant increase in high-grade aggressive tumors. Based on the possibility that the drug might be promoting aggressive disease, the FDA issued safety warnings for these medications in 2011. Subsequent analysis provided a physical explanation for these results. Finasteride shrinks the total volume of the prostate by approximately 50%, which creates a phenomenon called detection bias. In a full-sized prostate, a biopsy needle is less likely to find a small, high-grade tumor. By reducing the size of the gland, the drug makes existing tumors easier to target, increasing the detection rate of cancers that might have otherwise been missed. This shows how raw clinical data can lead to false conclusions when the physical changes of the organ aren't accounted for. If the drug is safe and effective at reducing low-grade cancers, it seems logical that it should be a standard preventative treatment. To find the final answer, urologists look at the most definitive metric in oncology: overall survival. Long-term data shows that after 15 years, the survival rate for the finasteride group was 78%, while the placebo group was 78.2%. Despite reducing the number of cancer diagnoses, the drug had no impact on mortality. Because the drug offers no survival benefit and carries risks of sexual dysfunction, medical guidelines now suggest against using it for routine chemoprevention in most men. There is currently no pharmaceutical shortcut for preventing prostate cancer. Until a new generation of agents arrives, the only verified ways to modify risk remain cardiovascular health, nutrition, and lifestyle choices.